arts. So it is a pleasure for us to have today in our Bar-Ilan University Vision Science Seminar, Professor Jorge Almeida from the University of Coimbra. I hope I'm saying this correctly. Professor Almeida did a 5-year BA in psychology at the Faculty of Psychology and Educational Sciences in the University of Lisbon, where his thesis was about cognitive and neural organization of knowledge about conspecifics and was done under the supervision of Leonel Garcia Marquez. At Harvard, he did a master's in psychology and then continued to a PhD in psychology in cognition, brain, and behavior. His dissertation, titled Unconscious Processes Reveal Different Circuits for Visual Object Recognition, was supervised by Alfonso Caramazza and Ken Nakayama. He did a postdoc at the Marie Curie, as a Marie Curie Fellow in a Welcome to Portugal program at the Center for Research in Psychology in the Faculty of Psychology in the University of Lisbon. Today, he is a professor at the Faculty of Psychology and Educational Sciences in the University of Coimbra, Portugal. He is the head of the Pro Action Lab, which focuses on investigating perception and recognition of objects and actions. Specifically, his lab currently investigates object recognition and the representation and organization of object knowledge, connectivity constraints on neural processing, representation, and organization, and neuroplasticity in special populations as deafness and aging. And this is done using a multi-method approach, including fMRI, neuromodulation with TDCS, visual psychophysics, and advanced analyses as machine learning, MVPA, and RSA. He published more than 60 manuscripts, many of them in high-profile journals as PNAS, Current Biology, Communications Biology, Nature Protocols, J Neuroscience, Psych Science, and many more. He gave dozens of invited talks across the world about his research. His research is funded by prestigious ERC grants as well as by additional ones. He has received multiple awards and honors, including the VSS Young Investigator Award, the Young Investor Award by the Portuguese Psychology Association, and many more. So I will stop here, and I'd like to welcome you, George, to our seminar to hear you about your exciting research, and thank you so much for joining us today.
Oh, thank you so much. And thank you all for being here. This is really, it's great. I was just telling Sharon that this is an opportunity for, in a crazy world that we're living, it's an opportunity for sharing science and sharing and friendship also. So thank you so much. Thank you for the introduction. Very kind words. So my work in my lab focuses on the organization of object knowledge in the brain as a proxy for the way in which we recognize objects. So object recognition probably follows from the way in which, or partly at least, the way in which information is organized in the brain. What I'm going to be showing today is what I think is a novel understanding of how information is organized. It's part of my ERC, which is called Content Map. So without further ado, I think, as I said, the topic is how do we recognize stuff in our environments? How do we recognize a key in this huge set of keys? I mean, everybody went through something along these lines, perhaps not as cluttered, but many times there's many keys. Which one is mine? Which one is yours? How do we recognize that this is a glass? And this is actually a glass of water. Unfortunately, I'm still able to do this. How do I recognize my daughter at a ballet show when every other little girl is dressed the same with the same hairdo, and I still recognize my daughter, fortunately? How do we do this? How do we go about the world and do these kinds of things? Well, I think it's interesting because whether it's a simple or complex process, it does seem automatic. It does seem, it's ubiquitous, and it does seem automatic. So, we see an object, and almost instantaneously, we get an answer. This is a glass of water. Now, despite this feel of automaticity, this is one of the most complex things our brain does. And one way to show you this is that, in fact, the process sometimes fails. And when it fails dramatically. So, for instance, this is the case of a patient who was studied by Alfonso Caramazza, patient EW. And patient EW was not able to name living things. So when shown this image of a whale, she was not able to say whale, but she was very good at saying that the other image is a set of strawberries, a group of strawberries, or a set of strawberries. She was also, when asked to say whether the sentence made sense or not, sentences related with living things, with animals, things like do whales have feet, she was not aware of. She was not able to answer, whereas she was okay with answering semantic, conceptual questions about other objects. In fact, she was essentially, when she thought about animals, she said, I can't even think about an animal. I don't know what it is, and I don't have the knowledge about it. Now, many of you might actually do work on cognitive neuropsychology, so you know this is not an individual case. There's many, many cases along the line, some of them exactly like patient EW, where you have a deficit for a particular category, in this case, animals, in the context of normal performance on other categories. But there's also opposite deficits where you have we're completely okay with knowledge about animals and you're able to recognize animals, but you're not okay with recognizing, I don't know, manipulable objects or fruits and vegetables or faces, etc. So we know that when the process fails, what seems to be easy turns out to be dramatic when it fails. But we don't need to go patient work to show that this is a complex process. I mean, I think, again, this set of images that I'm showing you here just tells you how complex the process is. I mean, we're able to recognize that there's a glass here with a tall glass in the middle of other glasses. They all are glasses, but we're able to say that that particular one is a glass, irrespective of whether it's made of glass or it's made of plastic or paper or metal. They're all glasses, and even worse, if the same glass is actually presented in different orientations, you can still, and the retina is going to be very, very different, you can still say this is a glass. So the actual complexity of the stimulus tells you really that the process is super complex. Okay, so, but I don't have to sell this to you guys. I guess you all know this is a super complex process to recognize an object amongst many others and amongst different types of visual information. And so I guess this is one of the reasons why a lot of us work on object recognition, trying to understand how we go about the world, how can we do this seemingly in such a busy way. And I think there's some assumptions that are shared by many, as I mentioned in the beginning immediately, which is I think many of us believe that understanding object recognition requires understanding the organization of knowledge. That is, our representations are organized in the brain is going to be very, very informative as to how our system goes about recognizing objects. I think this is a shared assumption. And again, there's a lot of work that has been out for many, many years on the organization of object knowledge. I'm just going to go through a little bit of it, but you guys know, I'm sure, a lot more. So for instance, there's data from humans and non-human primates that points to the existence of patches, areas, clusters, whatever you want to call them, but particular parts of the brain that care about particular categories of objects. And these include a subset of objects, a subset of categories, not all of them, but say faces, animals, body parts, places, manipulable objects, and this is where the assumption is, and this is where the consensus is, and then the question of why you have this patchiness perhaps is not as consensual, but you do have people that would say, look, these are really sharp functional divisions that reflect true domain distinctions. Others would say, no, not really. It's not really about this sharp distinctions. It's about similarity over particular dimensions or a particular dimension. I'm not going to talk too much about these two possibilities. This is the honest scope of this talk, but let me go a little bit further on what people have done in terms of the organizational knowledge. That I said, this work has led to the understanding that there are certain topographies. I would say they are coarse, but there's large organizational mosaics, large organizational properties of how information is represented in the brain. And so this includes, as I said, straightforward domain specificity, where you would say, look, really, you do have sharp distinctions. You do have areas that care about particular types of objects or processes. You have dimensions like object size, for instance, great work by Tal Conkle on saying, look, these topographies follow this real object size. So it's another possible topography. There's other work, also very beautiful, here by Arcaro, but actually before that, Levi and Malach and others have been talking about this, that you do have visual biases that probably influence the way in which information is organized in the brain. And you might have proto-maps that are related to these kinds of visual field biases from periphery to the center, and those are related to particular categories. And that's why you see certain mosaics, certain topographies. There's other topographies that have been out recently. So for instance, the idea that you have curvature or spikiness. And this is very interesting. You guys are vision scientists, in a way just like me. So curvature is a major feature of visual stimulus. So perhaps this organization by curvature, by spikiness is an interesting way of also carving out the neural space that we see that seems to be dedicated to object processing. And as I said, there's probably many other solutions to this patchiness. And I think most of them is going to say the same thing, which is with different stories, but same thing. Very large organizational mosaics, coarse topographies, non-continuous topographies, suggesting that you have patches of parts of your brain, that are representing particular aspects. In my view, and although I've also contributed to that, in my view, this is probably, it's important as a starting point, but it's not fine enough. You need to go finer. You need to start looking at what is happening within those topographies and trying to find more fine-grained organization of object knowledge. And so this is one of the things I'm going to try to show today, is that we need to look at tighter, more fine-grained topographies. Now, another assumption, this is probably not shared, and it relates to this previous part of trying to find finer topographies, is that perhaps a way to start this is to look at how the brain organizes other information. What are the kinds of strategies that the brain uses ubiquitously? And I think the best known organizational principles happen to be in sensory motor cortices. And so we probably need to go and try to understand what's happening in sensory motor cortices, and then try to see whether we can relate those to what's happening for a higher level or mid-level type of processing. And so let's look a little bit again. I mean, this is not, I haven't done this. Many of you, many other people, this is, I'm on top of giants here, on the shoulders of giants here. That people have shown, obviously, that you have fine-grained organization when you go into sensory and sensory motor cortices. So I don't have to tell you, but in vision, you have multiple dimensions that are represented. You have stimulus location, you have orientation, tilt, and you have a topographical, or a fine-grained topographical organization, where you have this Contentopic map that follows the position of stimuli in the visual field. And you all know this, you all know this topographic map, so I don't necessarily have to tell you them, but you can see here that eccentricity maps continuously in the way in which the brain responds to stimulus. So in terms of vision, you do have one, multiple dimensions, and two, stimulus location and orientation and others, and two, you do have a topographical organization where proximity in neural space follows proximity in a particular dimension, in this case, location in the visual field. The same thing for auditory processing. I'm going to skip this, but the same thing for auditory processing. You also have multiple dimensions, and you have a topographic organization. Again, proximity in neural space follows proximity in a particular sound-related dimension, in this case, tone. And you also see that in motor cortex. You see that in somatosensory cortex. And so, essentially, what I'm trying to suggest is that, from the inspection of low-level sensory motor information, you do have multidimensionality, and you do have topography as the main principles of organization. And so the question becomes, should the brain treat higher level objects, higher level information, the same way as it treats sensory motor information? My answer is, I would say yes. There's no reason for the brain to assume, to know that something is different. So what probably we should look into is whether we're using the same strategies to organize and represent object level information as we use to represent and organize sensory motor information. And so perhaps our object knowledge is also represented in a multidimensional way, and perhaps it's also organized in a topographical map, what we are going to be calling Contentopic maps as an analogy from a Contentopic map. So Contentopic maps is what I'm going to try to show you today. So let me go through a roadmap of my presentation. The question then is whether knowledge of objects is represented in a multidimensional way, it is organized in a topographical way also, in as much as sensory motor information is organized this way. And I'm going to try to show this first extraction of key object related dimensions and whether these dimensions are interpretable, then whether these dimensions are neurally and behaviorally a little bit behaviorally relevant. And finally, I'm going to tackle this issue of topography, true topography, right? Very similar to what happens to early visual cortex or early sensory cortices. Okay, so let's start with the extraction of key object dimensions. One of the issues, I guess, when we start addressing these topics is, unlike sensory processes where the dimension is rather, perhaps I would say, rather obvious. For vision is location, the visual field is you have a sensory organ, the retina, that follows that. And so the dimension is sort of given to you. When we think about objects, that's not true. That's not true because there's many dimensions, probably many of them are correlated with one another, and they are not as, if you will, observable. So one way to think about this is that the way in which we organize information about objects is actually or follows a 3D space. And this 3D space is going to have dimensions that organize, or actually multiple D space. And these multiple D space will have dimensions that organize the space. And so what we want to do is we want to extract these dimensions from the way in which people think about objects. Again, this is not me, this has been done before in many other fields, personality, etc., personal psychology. But one option then is to try and understand how people make decisions, judgments on objects, and try to take the organizing dimensions from the way in which people organize their judgments. And so, so assume that there's an ND space, and within this ND space, distance between two concepts is going to be related to the types of judgments that people make in terms of similarity of those two objects. And so what we've done is exactly this. We've selected a set of manipulable objects. Why manipulable objects? Because we know a lot about these objects, because they have many different types of information you can play with, and we know the neural responses to manipulable objects. So we selected a large set of manipulable objects, and what we did was we tried to extract the dimensions that are underneath the way in which we think about these objects and the way in which we judge similarity in these objects. So we started by asking a bunch of participants to, in this case, we use a technique that's called the piling technique, it's been used before, where people pile objects according to their similarity. And we asked them, we used words instead of pictures because we wanted to avoid particular, idiosyncratic similarity. We wanted conceptual similarity. And people were required to sort objects according to the similarity in 3 different types of information, independently. We asked them to do it by similarity in function, by similarity in manner of manipulation, and by similarity in vision. So we would have essentially 3 related judgments per person. They're independent, but they're related in function, manner of manipulation, and vision. So what we did then was we used non-metric multidimensional scaling to actually extract the latent dimensions. And we can discuss this later. I'm not going to focus too much on this, but we got a set of dimensions, a group of dimensions per knowledge type that we probed. So a set of 4 functional dimensions, 6 manipulation dimensions, and 5 visual dimensions. And we used regular criteria to select these dimensions. One thing that's important to know is that this is what multidimensional scaling gives us dimensions, and these dimensions actually allow us to think about the amount, quote unquote, of dimensional value that each object has. It's going to be very important for later. But then you might ask, what are these dimensions? So one thing we want to do is try to see if people could interpret these dimensions. And what we ask participants to do is to give names to potential the dimensions we were showing. We would show 10 objects that were on one of the extreme of one of the dimensions, and 10 objects that were on the other extreme of the dimension. And we asked participants to tell us, look, what could be the major difference between these 2 sets of objects? And we asked participants, they could put a bunch of labels, and in the end, we selected the most consensual label. And so I'm just going to show you examples here of dimensions. So for the manipulation information, we got as the first dimension strongest dimension, whether objects were grasped with a power or a precision grip. And this is actually interesting, given that this is the kinds of things that people would think are important for manipulation also. The second dimension will be a dexterity force, how much force you have to apply, and so on and so forth. For vision, this is very important. For vision, we had two main types of dimensions. One of them related to material, one of them related to shape. That's interesting because we know that the brain actually treats these two kinds of things separately. More in LOC for shape, more in medial aspects for material and for texture. And so the first dimension is whether it has metal. Second dimension is whether it's elongated or round, etc., etc. And for function, we got essentially a couple of things related to location where things are, and also the actual function. So kitchen versus sports, etc., etc. Now, I know this is a weak way of showing that these dimensions are real. Obviously, we are able, as I said, that is admittedly weak. It doesn't tell us much about the importance of these dimensions, just tells us that subjects are able to name whatever we ask them to name. And so we then said, okay, fine. So we have to go and look for to show that these dimensions are actually relevant. But let me just tell you before we go there, as a summary, we obtained object-related dimensions. These dimensions have been obtained from participants' judgments and understanding of what's happening that surrounds them. And sorry, there was a message I just wanted to see. But it's important to note these dimensions are noisy, okay? And especially the names that were given by the participants are to be taken with a grain of salt, right? I mean, yeah. So just bear that in mind that these dimensions are noisy. It is part of MDS, and obviously there's a high stress value, perhaps, but there's stuff that we're not explaining in terms of variance. And also, as I said, these are just approximations. And so when we say power versus precision, please keep that in mind. This with names given by the participants. Okay, so let's go for the second part, which is trying to show that these dimensions are relevant. It is that they somehow are important for the way in which information is organized in the brain. And so what we did here was we wanted to show that essentially that the way in which the brain responds to these different AT objects can be explained by their values in the different dimensions. Okay. So the way we did is, well, let's put this let's put participants in fMRI. Event-related design, we show our AT objects many times. Every time we show that there's going to be response, there's going to be, and so what we know is that there's going to be different responses for different objects in each particular voxel. And we're going to try and see whether the variation in response actually can be explained by the variation in the actual dimension. Remember that I told you that with multidimensional scaling, every single dimension that comes out actually means a value per object. So we know the amount of power precision, whatever that means, for each particular object. And so we can actually go in and say, look, we can use as a predictor these values per dimension and try to see whether the variation of the values per dimension, that you can see here in one of these, explain the actual variation of responses. Okay. So this is a parametric analysis. For those of you that know, it's parametric over fMRI, again, typical. And so we just want to see whether our key dimensions can explain the variation of signal. Okay. And so I'm going to go relatively fast here and just to show you that, in fact, we do find areas that we would assume would be important for the different types of dimensions. So let's start with the functional dimensions. What we're showing here in different shades of blue, bluish, is how different dimensions, functional dimensions, explain the signal changes for the AT objects. So for instance, you do see PMTG, PMTG and LOC, which are areas known to be associated with function, work of Moritz, Moritz Wurm, Fonseca Carmazza, Angela Galin now, Paul Donning, etc. People have been showing this for sure. And they respond to different kinds of actions at a level, perhaps at a more conceptual level, which is interesting. But we also see vectoral stuff, more, if you will, more some interior vector stuff, but more interior vector stuff, which is also known to be associated with action and with function in this particular case, the work by Marius Piller and others. And we also see IPL. So we see left IPL, which is important. We know it's related with action and function. And so function at a higher level. And so this makes sense. What we're trying to show here is that, in fact, these dimensions can explain part of the activation for these 80 objects. This is for function, but we also have for manipulation. You can see manipulation is more dorsal occipital, more early parietal cortex, more parietal cortex happening here. But you might also see that there's stuff in ventral stream. And by the way, this is something we've been showing, but not just us. There's many people now showing that there's activation for manipulation related information in ventral temporal cortex, namely things related to functional grasps that seem to be related to activation in ventral temporal cortex. But here essentially I want to show that there's, when you go into manipulation, there's more of these dorsal occipital areas responding, but also more of these parietal regions responding to this manipulation. Dimensions. And finally, the visual dimensions, what we can see, I think what's very clear here is that you have a lot more responses, or in fact, these dimensions can explain a lot more of the responses in ventral temporal cortex. The black here, sorry, I forgot to mention, the black here means that there's many dimensions that explain the results there. And so the visual dimension, a lot of the visual dimensions explain a lot of the ventral temporal cortex, which is expected. Okay, so I guess what I want to say here is that again, we know these dimensions are to be taken with a grain of salt, but they do seem to track neurally, at least, and I'm going to show you next, that also track behaviorally, track neurally to the object processing apparatus that we have. And so just very quickly, we also do this behaviorally, and in this particular case, we wanted to train subjects, participants, to learn how to categorize objects according to the values of these objects in the dimension. And so we trained objects in the set of we trained participants in the set of our objects, so we trained them in half, or almost a bit more than half of our objects, 50 of our 80 objects. They were trained to do classification. So horn, so for instance, pepper grinder is closer to knife. Knife is closer to pepper grinder than to horn, and dart is closer to horn than to pepper grinder. So they were trained to these kinds of decisions. But what I really care is not this. What I care is what happens when we then present the remaining 30 objects that were not trained on. Is there generalization from their learning? And what I want to show you here is that, yes, there's generalization. So these graphs here, both this one on the left, on the lower left corner, but the other ones also, are all graphs from the 30 untrained objects, showing that there has been very strong, by the way, very strong generalization of the learning to untrained items. That is, participants actually learned how to classify objects from the dimensions we've selected. And just to be sure, this is all the other dimensions. So there's always, as when you start going to later dimensions, which are weaker, you start seeing a flat curve here. So there's less generalization in the later dimensions. And when we use a random sequence or when we use lexical frequency, you start seeing flat also. So there's no generalization, there's nothing to generalize. And there is generalization when the learning is from dimensions that actually makes sense. So I guess what I'm trying to say here is that, in fact, these dimensions are neurally and behaviorally relevant. And so, in a way, multidimensionality is a hallmark of object recognition also. It's not just us saying that, many people saying that too. Martin Niebert, etc., also saying this. That part is not. It's novel because it's a within category demonstration, but there's other people saying the same. But now let's go to the final part of my presentation, which is, okay, we've shown that there's multidimensionality, but is that topography? And continuous topography, just like retinotic. Do you see that when you start going and working with objects? Okay, so to test this, we first selected two of our dimensions, the first two-dimensional manipulation. We are working now on other dimensions too, visual dimensions, etc. But here I'm just going to show you the first data set that we have, which is with these two manipulation dimensions. Again, the labeling, that doesn't mean taking a grain of salt. The labeling is power versus precision and dexterity versus force. Those are the two first dimensions. And I just want to remind you that because we use multidimensional scaling, we do have a value per dimension per object, which means we can rank order these objects by the dimensional values. And this is important for what comes next. Okay, so our understanding of going about this question, what we want to do is that we should use what is typically used for red knot. Okay, that is, in this particular case, a population respective field analysis type paradigm. But instead of changing the location of a stimulus in the visual field, which is the dimension that matters for visual cortex, we're going to change the dimensional location on the actual dimension, right? So how can we do this? Well, we can go from low dimensional values on the first dimension at what we call M1, so power precision. We can go on low dimensional values, which would mean power grasped items. And so if you start showing power grasped items, those are in the lower part of the dimension. Well, then you can keep on going and start showing the next items, and the next items, and the next items, and the next items, always in the middle. So there's no red knot over here. And eventually you're going to end up in the last part on the other extreme of the dimension. And so if you progress this way, you're going to go, we did it clockwise and counterclockwise in the same participants, so that's taken care of. We will start with one part of the dimension and present objects from that part of the dimension and keep on going until we move from the beginning of the dimension to the middle of the dimension to the last part of the dimension, and then we restart. So there's a bunch of cycles. So what are we doing here? What we're cycling here, instead of cycling the visual location of the stimulus as we do for ethnotopy, we're now cycling the different levels of the target dimension, right? So we're showing objects from different parts of the target dimension in a sequence, just like we do for ethnotopy. And so, by the way, what we did was for every cycle, we select 40 of the items, we present them in order, then the next cycle, we present the remaining 40 items, and then we do this again, okay? And we do it 6 cycles per run, and 6 cycles per session, and this is 8 participants, and each participant is there for a long time. So there's plenty of things. So then so what do we do then? Well, then what we have to do is what we have done for retnotopy also, which is create models with a particular preferred location in dimension and a particular tuning width. But this time, again, we're not looking at location in the visual field, but location in the dimension. So this is actually a 1D Gaussian, not a 2D Gaussian, but a 1D Gaussian that goes through the particular dimension we're focusing. So we can create models that care about this part of the dimension, one of the extremes. And this would be the preferred location, right? But then the width can be slightly different, right? And so we can keep on creating different models for different parts of the dimension. And then we're going to test it and see whether we can model the data. So essentially, what are we modeling? How cycling through different dimensional levels is going to elicit different preferred responses in contiguous parts of the cortical surface, okay? Just like we do for retnotopy. Okay? And so this is what we do. And I hope this is clear. I think it's very clear because it's exactly what people have done with retnotopy. So same thing, except that we're looking at a dimension, it's a 1D Gaussian, and we're looking at a particular dimension. Okay, so is object or knowledge organized topographically by these dimensions that we selected? So the first thing I'm going to show you is that our 1D model does capture signal function. Okay? Yes, it's in occipital regions, maybe some temporal, some parietal, but it's mainly occipital regions, it's true. But it does capture. And so, for instance, in your examples that I'm showing here of voxels, where the response is really following. So this voxel 1, there's a voxel that in part A of the figure is a voxel that really follows the cycling of the dimension. Okay, so by the way, in red is the model data, and in blue is the dots, obviously the real data. And so in some of these voxels we model very well the data, in others obviously we don't. This is a typical thing. What we've done to make this easier is actually create ROIs. I know it's not perhaps we'll be ready for another way of doing it, but because what we'll do next, we have to create ROIs. So what we did was create ROIs where all our participants show a good percentage of signal capture. We're capturing signal fluctuations. And this means that we now have two ROIs bilateral. One that we call dorsal occipital, which includes V3A, but also includes V2, etc., etc. I'm going to show you later with an atlas, so you'll see that these are a bit posterior. I do acknowledge that. And we also have a ventral ROI that we call VLCC, which includes the lingual gyres, but also, as I said, V2, etc., etc. Now, these are the regions that were very consensual across all the participants, but note that there's for some participants that I'll show you later, there is parietal activation too. Some others have more ventral activation, etc., etc. Okay, so and what I wanted also to show you that we also obtained the control region in the frontal cortex, just to compare. We also want to tell you that we tried alternative models. This is a 1D Gaussian that we've used, but we could use a linear model or other possible without only the location, for instance, without the tuning, and our model always exceeds the other alternative models. Okay, so I'm going to focus now on a different thing, which is we've shown that we're able to capture signal fluctuation, but that could be completely random. I mean, it didn't have to be continuous. It could just be that, there's pockets of this part of the dimension, that part of the dimension, each voxel could be completely different. There's no reason why this should be continuous. But that's what we really wanted to look at. We wanted to look at preferred location. That's why we came up with these ROIs, by the way. And what I'm going to show you here is that, in fact, we do get continuous maps for both the first dimension and the second dimension. Just going to show you the first dimension, but let me show you. So when you start showing objects that go from high dimensional values, in this particular case, precision grid, and you start moving to more, still precision, you start moving in a dimension, you're going to start to see a buildup of responses, a buildup of the most preferred location in the dimension. That goes from, if you will, from inferior to superior in this dorsal occipital area. And so this is a first demonstration that there is a topography that follows the dimensional values. And you can see here the average maps. So you can see that there is a progression. We also then looked at actual orientation and see if there's a particular orientation for each participant individually, and there is. And so let me move now to trying to test this topography. So the first thing we wanted to show is, look, well, we got these maps. They seem to be topographic. They seem to be continuous. Is there consistency across participants for each dimension? Now, if there is, we should be able to predict an individual map from the average of the other maps. So if we take the map for power precision of participant 1, we should be able to predict that participant's map from the average of the responses to dimension 1, so power precision for all the other participants. So this is a live one out cross-validation situation here, where we correlate the preferred location map. It's a preferred location map correlation from the left out participant with the average of the remaining participant. And so we do get two things here: R squared and slope. Okay, so it's a linear correlation. R squared means whether they're correlated or not. Slope actually means whether they're in the same direction, which is important because they could be correlated but completely opposite directions. They're not there. And so as you can see here, both in R squared for M1 and M2, both in R squared and slope, is that we're actually able to predict individual maps from the average of the remaining participants, and the slope is positive. That is, the orientation is the same. This is obviously very interesting, and you can see here the average, so you can see that they follow the same orientation, just to show you that they follow the same orientation. But now you might ask, well, maybe whenever you present stuff in a particular order, whenever you present stuff in order, you're going to get a map of this sort. So the next step, obviously, is to show that these maps are specific for each dimension. And so we're doing is the same thing. It's a leave one out, one participant out cross validation, where we are going to see whether the map of one participant for dimension 1, power precision, is explainable by the map of the remaining participants in dimension 2. Okay. Is it the case that these dimensions are independent, or are they very similar? And so if they are different from one another, we should not be able to predict Contentopic maps for, say, power precision on one participant from the Contentopic maps for dexterity and force from the remaining participants. Again, this is cross-validation one out. And what I show you here is that no, we cannot. So they're very different. There's a difference in R square for both R1 for both M1 and M2. These are significant differences in a way, so we cannot predict the map of one subject in dimension 1 by the remaining subjects, the maps of the remaining subjects on another dimension. So these dimensions are specific. And again, you sort of can see it here, actually. I mean, you can immediately see it here that if there's consistency for M1, for instance, M1 and M2 are very different, so they would not be able to predict one map from the other. Now, you guys are vision scientists, and you might be asking, it's fine, but your areas are very posterior, so perhaps your maps are not dimensional maps, and they are just an unfortunate correlation with, say, eccentricity, or nothing. And I'll discuss this in a second, because I'm not suggesting that there's nothing retinotopic here. That's not what I'm saying at all. But what I want to show is that these maps are not reducible to low-level properties of the kind that drive early visual cortex. That is, they're not reducible to eccentricity or to, if you will, retinotomy. And so the way we went about to do this is two ways. One is to use retinotopic mapping that's available in the literature. So we took the Human Connectome project, this beautiful, beautiful retinotopic map, very, very well done, 70s. It's a very detailed map, and which you can see here in D. And we would take those maps and see whether we can predict individual maps from these HCP maps. That's one option. It's not my preferred option because this is the surrogate data. So can we do it with our own data? And what we did was we actually went back on our data and said, fine, I mean, there's variations of the stimulus, and we might actually be able to get a retinotopic map from our own stimuli. And that's what we did in C. In C, we look at the energy, the contrast difference by location in the visual field, and we compute a traditional, regular retinotopic eccentricity, in this case, eccentricity map. So that's what we have here for our own participants. And then the question is, can these, and this is important to say, these maps come from the same exact stimuli as the maps from A and B. Okay? Exact stimuli. This has to be very clear. And so if our maps in A and B, that is our maps for M1, Contentopic maps for M1 and M2, are reducible to eccentricity, then we should be able to predict the maps. As you can imagine, we cannot. And so here's the response. Very clearly, we cannot predict our maps. Again, with the leave one out cross-validation design, we cannot predict our M1 and M2 maps, that is our power precision and dexterity and force, again, grain of salt, maps, Contentopic maps from the retinotopic maps, whether they are derived from our own data, our own stimuli, the same stimuli that actually created the A and B, the maps from M1 and M2, nor from a set, very detailed, very strong retopic map that we obtained from HC. Okay, so this is that I'm going to show you. I'm going to go into the discussion. I should have said you shouldn't drop me if you have any questions. Sorry about that. I just started speaking in. Sorry about that. But I think what we've shown here is that multidimensionality is also a hallmark of object processing. And other people have shown this, as I said. Martin Niemann has shown this. Chris Baker has shown this. We have shown this. Other people have shown this. So it's not necessarily novel, but it's an important aspect. Is that multidimensionality is a hallmark of object processing. We're able to extract object dimensions. People actually use them to guide their behavior, and they actually are also relevant neurally. But more importantly, we obtained contentopic maps, which is a novelty here. So the idea is that mentally traversing a target dimension leads to continuous and greater progression in neural responses, just like mentally traversing or actually traversing a visual field will also lead to a progression in neural responses, if you will. So a true topography. And these topographies, I hope I've shown you this, are continuous, okay, that they progress through the cortical surface. And that they do so as we go also through the dimensional values. They're consistent across participants. We've shown that with the cross-validation. They're specific for each dimension. We show that when you try to predict one map from one dimension from another dimension, you cannot do it. And they are relatively independent of low-level properties of the stimuli. So we've shown that, in a way, retinopathy by itself cannot explain the results that we obtain. And so the answer to the question of are these just low-level properties? I would say no. But first of all, it's important to show, and I'm completely aware of that these are very posterior areas. Maybe you were expecting a bit more interior areas. There's reasons for that, perhaps. I don't think we're talking about high-level content here. I think we're talking about mid-level content. I think we're talking about the types of information that are in between the concept of a hammer and the fact that we have lines and corners that we do as we work in early visual cortex. That's what's happening here. But it's also important that I think this is, in a way, if you will, independent from a low-level visual properties, but these are strong retotopic areas, very strong retotopic areas. Okay? So it suggests there has to be a relationship. However, I think it's important to note that it is possible, and people have shown this many times, that in the 2D cortical sheet, you can actually have different topographies that coexist. This is important to say. But these contentopic maps do lay in part superimpose into strong retotopic maps, and I think they are related. And honestly, in the real world, there are certain aspects of eccentricity that correlate with the different types of objects. So a power grasp object is typically a more elongated object, and so it does correlate with eccentricity. So, an elongated object is probably going to be more in the periphery, especially when you focus, when you foveate the object. And so this might be actually related. And also, you have to maintain attention. We've shown that before. You have to maintain attention towards both the functional tip and the handle. So eccentricity might actually be a very important thing here. Moreover, hand pre-shaping and finger placement will probably require low-level information, location, etc., etc. So there's no reason why we would suggest that contentopy and retinopathy are not related. In fact, I think they are. And it's also interesting that this reconciles a lot of the work showing that early visual cortex actually responds to different types of grasps, and it does so in the absence of vision. So it's not a visual response. This is the work by Simona Monaco and others that seem to suggest that this is the case, and you actually can decode the grasp type from early visual cortex. So contentopic maps could be like sort of proto maps for mid-level information that allows for this hand shaping and finger placement and etc. Some of the motor stuff that we care about. So are these mid-level properties? Well, I think they are. I think they are intermediate representations that bridge early visual properties, these lines and corners, into these complex object representations. Okay, a hammer, it's whatever, right? And these mid-level representations are important we know they're important for visual motor objects. It's beautiful work by Gilles Van der Skorp on a patient that has or participant that has issues with this mid-level perception, and she does have issues with perception of objects and things. And I think also that one of the interesting things about these Contentopic maps is that they no longer focus on spatial coding. They're probably more focused on object feature mapping, on these parameters, and this maybe you now start to impose an object-centric frame reference and not so much an egocentric frame reference. And speculatively, and this is very speculative, I think what's happening here is that these Contentopic maps will then aggregate into and will aggregate and will connect to more interior parts, creating these domains, these mosaics that I was telling you in the beginning. So these coarse topographies, I would say, we're testing that right now, are related to this Contentopic map. So this domain specific that we see downstream might actually be related to this Contentopic mapping, these mid-level properties that we have. So as a conclusion, I think that what we showed here is that one of the strategies that the brain uses to store information, represent information, especially sensory motor information, is also used for other stuff. So this is a ubiquitous strategy that the brain uses. It tends to represent the information in multiple dimensions. It tends to represent it in a topographic way. There's reasons for that. It's not incidental. This organization is not incidental. It's widespread. It's related with less metabolic or reduced metabolic cost. And I think this is the most important thing. It relates to what I was just saying before. It allows for easy readouts of the information that then can be built up into another map, and then build up into another map, and then build up into another map. Anyway, I want to thank my lab, the Proaction Lab, and especially these people here. Alessio Fracasso has been a great collaborator. This is the work of Steph Christensen, Zohar Tal, but also there's other people involved, Frederick, John, Daniela, the remaining production videos. Thank you all. And before I leave, I just want to direct you to this meeting that we have next. We've been having it every September. So I recommend you to go. These are the speakers for the next edition. By the way, this is the first time that I'm showing you the speakers to everybody, so you guys should feel honored because you're the first ones to know. And I hope you guys can come. And thank you very much for your attention.
Wow, thank you so much. This is a really great talk, and I want to invite everybody to unmute yourself, and let's give George a big applaud. And I have to say that Mel apologized. He was on a train and the connection was weak, so he said that he would be watching. He would hope to see the recording because he had to. He was disconnected. So yeah, this meeting looks wonderful, but are you okay to take questions about the
Hey, more than okay.
Okay,
Welcome
lovely.
questions.
So actually I have a lot of them, but if other people have questions, feel free to raise your hand. So one thing I was thinking about is if you examined or if you thought about examining, doing tractography and DTI and trying to see whether, there's some arrangement in the connectivity downstream through maybe the ventral and the dorsal portions that you actually got are connected in some, I don't know, some linear progression, or maybe they are connected to other areas in some orderly manner. So, I mean, if this is really a dimension, it would be fascinating to see how the connectivity is arranged.
Absolutely. So sorry, I know.
Yeah, no, I've got a question, but I'll.
Absolutely. So tractography, no, but we are. This is the project I was telling you, sort of alluding, is that we're looking into connectivity, and yeah, connectivity between these maps and other areas, and see how these maps actually create other maps in more interior areas. So tractography, I never thought about it, mainly because these are particular. These are gray matter areas. It might not be so easy to sort of go into particular parts of these maps and say, okay, tractography from here to here. But that could be an option too. We do have data, by the way. And there's collaborations to be done here, but we do have data, plenty of data on DTI with participants that did experiments with these items. So we could actually do that.
Oh, you have the data. Wow.
We have DTI.
Oh,
Not
amazing.
with the maps, not unfortunately with the 8 subjects that participated in the content topic maps, but with the other ones that participated with the dimensional, with the parametric analysis. Then we have, for every single one of those participants, we have DTI. We have DTI measures. Yeah, so we could do that. But as I said, we are following a different approach on connectivity, mainly a functional connectivity. Not a typical one. We're looking at connected field approaches, trying to look more into the connected field type stuff, like people have done. Again, this is, I feel like I'm ripping off people that do retinopathy. So we're taking exactly what they did with, and now we're going to do exactly the same thing, but now with our maps. And so connected field mapping is one of the things we want to do by taking these maps and looking at how they interact or create or relate to something that's downstream. Yeah, but tractography could be an interesting idea, and perhaps we could share about that later. You're
Yeah,
muted.
that sounds really exciting. I mean, along these lines, and I see that Naama Friedman is here, and she's an expert in aphasia, or sorry, lexical issues and language. And I was wondering, if there's some overlap, because if there's some semantic or action-related activity, maybe there's some type of aphasia, or that actually mix these dimensions. There's different language-related deficits. Sometimes you can see deficits within some semantic zone, let's say, and it may actually correlate with the dimensions that you find. I don't know, but I'm thinking maybe
Yeah,
that would be really
yeah,
interesting to examine. Today with all these language models and everything, it would be really interesting to examine if these are dimensions that match some types of aphasia, or
yeah,
some of them at least.
yeah. So before I answer your question, let me give you a caveat here. Our experiment was visual.
Yeah.
And it was visual. It was visual without a task necessarily. And that's a problem for us, anyway. And it's a limitation. It's a limitation, and I think some of this, the reason why we have a lot of more prestigious stuff is also because it was just a visual task. Now, if you go into a motor task, we could start getting more parietal stuff also coming out, and that could be interesting for the question that you asked, because there's a lot of relationship between St. Broca's area, and our premotor, ventral premotor, etc. So there's connections that could be formed. Whether this is conceptual or not, this is what I was trying to say. I think this is mainly mid-level properties happening here. So it's a step in between concepts and low-level vision, which we'll know a lot about because it's very hard. John Wagemans is always saying that, look, this is very hard to go into this mid-level stuff, because what is that? But I think it's an interesting question. We are, as you might imagine, given that I'm working in Alfonso Caramazzo, we do love to test patients, and we do test them. And Alfonso is actually in Coimbra also, so Alfonso is working with us here, and so we are looking into patients, and not necessarily aphasics. This is not an area that I feel very comfortable about, but apraxic patients. And these apraxic patients could be interesting. Segue to the question that we asked. And I think this is one of the options to follow is, can we see particular deficits in certain aspects, certain of the dimensions, but not others, when we test the apraxic patients, for instance. When we test the aphasics, maybe, but I have to understand exactly what each aphasic patient can give me.
No, I'm just thinking, there's some or there's some aspects that people who can confuse a chair with a table, and all these kind of semantic related. Now, I know you're saying that this is visual, but maybe the representations, I mean, my thoughts are that once the representations are in adult life, let's say, or when the visual and the language and everything is kind of like interconnected. So I'm just thinking maybe there are, some ties between semantic and language and vision, even though it's mid-level. I mean, maybe I'm wrong.
Yeah.
I mean, it's
No,
just
no,
I
no,
thought
not a
that
problem.
I
It's
had.
an interesting question, yeah, for sure. It's an interesting question. It's something that could be tested, especially with the pricing questions for now, but eventually that. Yeah, for sure.
Yeah, it's fascinating. And one thing that I also thought about, but I'm not sure. Okay, how these regions, I mean, I assume that they're a bit more posterior than EBA would be, but do they overlap to some extent with EBA? And then,
Some of them, the more interior. I mean, some of them are LOC-ish. Um, Especially for M2, there's actually a snippet that we didn't analyze because it doesn't match with M1. It's more interior and more lateral. That could be around, yeah, potentially around there. But I have to say, as I said, these are more posterior than I thought they would be, and I think it relates with the kinds of stuff, well, also with the kinds of properties that we have here, but also with the technique. So I think if we're using more of action or more, maybe we'd get these areas plus a bit more parietal, for instance, and plus perhaps if we take a task that's more, I don't want to call it semantic, but more object related, maybe you start seeing a bit more, more, more interior into areas, as you were saying.
uh-huh.
Maybe. So I'm saying it's different maps, so perhaps multiple maps related to these topics.
Wow, this is really yeah, exciting work and really mind challenging. I'm also thinking about DF, how her lesion relates to your findings, and so a lot of directions and thoughts, and
Yes.
really beautiful work. Um, so I want to thank you again for this wonderful talk. Thanks so much for joining us. And this is your the last great talk of the first semester. We're just ending our semester here, and
Wow.
we'll continue in a
Wow.
month or two when the second semester begins. Oh, Lucia, I see you're here too. I wasn't aware.
Olá, Lúcia. She's
How
a
lovely.
country
Hi.
dancer.
Sorry, I just wanted thank you so much, Josh. That was really interesting. Lots of great food for talk. Sorry, Sharon, I didn't mean to
No, I'm
interrupt.
so
Thank
happy
you so
you
much for
unmuted
organizing the talk.
yourself. Great. I wasn't aware that you're here. Lovely, lovely to see you as well.
Yes.
You
Olá.
too.
Há muito tempo. Já nos vemos.
So thank you so much, and thanks everybody for joining. And yeah, have a lovely continuing of the year, and we will catch up soon, I hope.
Thank you
And
very
I hope
much.
I'll be able to come to Portugal for
Yes. Don't
yeah,
forget, beautiful
yeah,
workshop. Great people.
yeah, definitely. I mean, it looks amazing, amazing.
Okay. Thank you very much. It was very
Thank you very
nice.
much. Thank you.
Thank
Goodbye.
you.